Our next speaker this morning is Dr. Adviye Ergul from Georgia Regents University. She's going to talk about cerebral neurovasculature and diabetes, the good, the bad, and the ugly.
Thank you very much. Well, good morning. It's such a pleasure and honor to be here and to share this data with you. I look forward to your feedback. So as we know, brain is a highly vascularized organ with miles and miles of capillaries, and it gets almost like 20% of the cardiac output. And that's because, as Dr. Ergul also touched, it has no energy reserve. So it heavily depends on this constant blood flow, constant perfusion. And this is obviously a very tightly regulated function. And one aspect of the cerebral blood flow regulation is functional hyperemia. And it also is a good example of how the blood vessels in concert with the neighboring cells regulate blood flow to the brain. I see many of the investigators who have contributed to this field in the audience today. And here I share a slide from Dr. Flosa, and in which they pointed, if I see this thing correctly, use this correctly, that the signals that the active neurons signal to the neighboring vasculature through signals that involve the astrocytes, so that the blood vessels know to dilate and provide the blood flow where it's needed. But as Dr. Flosa would strongly argue, that this is not a one-way street. Actually, this communication is bidirectional, and we also the vasculature signals to the neurons to regulate this blood flow. So as you would appreciate, this would require a highly structured, highly organized vascular networks, as we also heard yesterday. And also you would appreciate like the disease states would and the metabolic demands associated with these disease states would affect this highly organized structure. And we know, and I think it was pointed out yesterday as well, that cortex would have more vasculature than what we see in the striatum or the white matter. And what I'm going to touch today is how diabetes might affect this system. We know diabetes is associated with a number of complications, including eye and heart and peripheral vessels and the brain. And all these complications have a vascular component to it. Vascular disease plays a very important role. So several years ago, we started looking at the vasculature in diabetic animals. And in these studies, we used what's called a Goto-Kakizaki GK rat model of diabetes. These animals are spontaneously diabetic, and their blood glucose is not severely elevated. So it's a good model of type 2 diabetes. And as you would see on the right, we have our diabetic animals, and on the left is the control animals. This is just now looking at the Pilser vasculature, like Dr. Faber nicely showed us. We noticed that the vessels in the diabetic animals, they're much more tortuous, and they have more collaterals. Now, then we decided to look in more detail into this and look actually in the parenchyma, and we used this space-filling model to visualize and quantify the vasculature. And here on the left, you see the control animals, and on the right, the diabetic animals. And we looked at the vascular density in various areas of the brain, and here is a representative from the cortex and striatum. And you see that the diabetic animals, to our surprise, they had the vascularization was much more increased in these animals. And we quantified this throughout the brain, And here what I have on this graph is actually going from front to back. And we focused on these areas because if we induce stroke in these animals, as I will share with you later, these are the areas where we see a lot of bleeding, and that these are the areas close to the infarct that we observe. And as you see in this graph, in red, the diabetic animals, in both the cortex and the striatum, the vascular volume and vascular density was significantly increased. And if we co-stained these slides with a CD31 or with leptin, actually we saw much more vessels that are not perfused with FITC, indicating there are also other new vessels being formed, but they are not yet perfusing. And when we looked at the parasites in a study in collaboration with Paula Duffy, actually we saw that the parasites are much less in our model. And in this particular experiments, we looked at it by just looking at the nuclei, where the endothelial cells are these elongated nuclei, and the parasites they have the round nuclei, and the parasite coverage was reduced in our diabetic animals. And then by doing a serial of analyses using these images, we are able to look at the microvasculature versus relatively larger, what we call macrovasculature, but they're still, of course, small, that we were able to quantify some additional parameters. So obviously the question is this something preventable? So we treated our diabetic animals with metformin, one of the most commonly used anti-diabetic agents, starting at the onset of diabetes. And if we do so, then we were able to correct some of these changes. In particular, we saw increase in branch density in our control animals. And this was not affected by the metformin treatment. But if you looked at the lumen diameter of these small vessels, it was reduced, which was increased in diabetes, was reduced with the treatment. And also the ratio of the overall, the vasculature was much more increased. And then we were able to restore this with metformin treatment. So what are the functional consequences? In these animals at this age, functional hyperemia is impaired. Dr. Adekola nicely alluded to this. We looked at we used the viscous stimulation approach. And as you see here, the diabetic animals in closed bars, they don't dilate as nicely as the control animals do. And their permeability is increased. And also, we also looked at novel object recognition test to kind of very crudely assess cognitive function, and we see that these animals there is impairment. Is this something treatable? Well, then we took our animals after they developed the disease, and then we treated them with metformin. And we used a 4-week treatment period. And as you see here, with the metformin treatment, actually we were able to reverse some of these effects even after the disease is established. Is this something specific to brain? We looked at skeletal muscle and also the retinas. In the skeletal muscle, actually we saw much less blood vessels. However, they were, as you see here, they were bigger, they were remodeled, but the density was less. On the contrary, just like is seen in diabetic retinopathy, we actually in the retina, the vascular density was increased. Is this something specific to the diabetic model we used? Well, we then used a mice model. We used the DB mice, and we looked at this. There were some differences in terms of the micro versus macrovasculature, but what we have seen is, again, these animals, the diabetic animals, DB mice, they have increased vascularity. So what are the potential mechanisms? These animals, they do have increased VEGF, and they have increased basal phosphorylated VEGF receptors. And we think that they actually, through peroxynitrite generation, they're mediating this angiogenic effect. Well, it also has another consequence, and that is if we stroke these animals, if we induce an acute ischemic stroke by occluding the middle cerebral artery and then reperfusing them, then these animals are more susceptible to bleeding. They develop what's known as hemorrhagic transformation, a complication of ischemic stroke. And what was interesting and challenging for us was that actually diabetic animals, they developed smaller infarcts, as you saw here. However, the functional outcome was much worse because all these animals had quite bleeding into their brain. And we looked at this by using different methods of stroke, either embolic or different occlusion reperfusion time points. At all time points with all the methods, we saw in our diabetic animals that they have greater bleeding into the brain. So that's why we focused our efforts on vascular protection strategies to reduce the injury and also to actually, hopefully, to improve the outcome or the repair after stroke. So neovascularization after stroke is important, and angiogenesis has been suggested in the peri-infarct area that is associated with better recovery. So that's why we also looked at angiogenesis after ischemic stroke in our models, and the data that I'm going to show you today actually from areas that are distal to the infarcted area that you see here on the left. And as I've shown you before, at baseline, our diabetic animals, they had greater vascularization. But now I'm going to draw your attention to the post-stroke period, and first looking at the control animals, that they have actually increased angiogenesis as compared to baseline sham animals. So indication of some reparative angiogenesis that had been reported before. An interesting point was that we also looked at the contralateral site, non-ischemic site, and we saw increased vascularity in the contralateral site too, so that ischemic injury somehow is turning on this angiogenic response in both sides of the hemisphere. What was interesting, though, after stroke, all this vascularity, I mean, we saw a huge vasoregression, mostly in the infarcted area, but even on the contralateral side in diabetic animals. And we don't know why that is. One thing we've done, now we took these animals, diabetic animals, and treated them only in the post-stroke recovery period. And I forgot to mention that these are 14 days after the stroke. So if we treated their blood them with metformin, regulated their glucose in the post-stroke recovery period, we were able to provide some protection against this vasoregression that we see. And also, obviously, vasculature is not alone. We looked at astrocytes, and we saw that there astrocytic swelling in the diabetic animals, which was prevented by the metformin treatment. And we also looked at number of processes per astrocytes, and we also saw that metformin treatment was effective in preventing this. We looked at functional recovery, and in this case, actually, just looking at the number of tests and resulting in a composite neurological score. And as you see here, all animals kind of, as you would expect, they had a low score after stroke, but then they recovered. Control animals actually did quite well, recovering all the way to normal. But our diabetic animals had their recovery. They did recover, but not fully as you would expect. We also used this novel object recognition that I mentioned earlier, and Dr. Ayodokolo also mentioned, and that this is a test that looks at the ability based on the fact that the animals would look for the new object if they remember it. So by using this, one thing we noticed was at baseline, our diabetic animals had actually lower recognition index. And they did recover, normal animals recovered some, but the diabetic animals, they had a very blunted response. So lessons learned, diabetes impairs reparative vascularization and causes vasoregression after stroke. So if you take a control animal and stroke them, you see reparative angiogenesis, which is good. But if you have diabetes, you have this dysfunctional new angiogenesis. Yes, you do have vessels, but they're leaky and they're not fully functional, which is bad. But if you put the together, actually you start losing these vessels, and there is vasoregression, astrogliosis, so, which is ugly. So more vessels, definitely it's not equal to better function. And now as we use therapeutic angiogenesis in diabetes, and especially after stroke, we need to be very careful. Microenvironment seems to be very critical for vascular integrity and restoration. And still, glycemic control is quite an effective strategy to prevent some of these cerebral complications. Well, I think a lot of challenges remain. How do we promote adaptive and reparative vascularization in health and disease? Is cerebral angiogenesis, I mean, is this something that is attainable? Is it always good? And for Dr. Miller, are there sex differences in this response? Our initial studies suggest that the females also develop this pathological neovascularization in diabetes. And how is function affected, neurovascular coupling affected? And what are the mechanisms just in diabetes? Is this a pruning defect? Is this a maturation defect? Or after stroke, what are the underlying mechanisms of vasoregression? Obviously, it takes a village to do all this, and I would like to thank my collaborator, Susan Fagan, who drew me into the stroke field, and to all my students and trainees and collaborators, and obviously NIH and American Heart, I have to acknowledge. Thank you.
Thank you. That was a great presentation. Questions?
Hi, that was Jim Faber. That was very elegant work, beautiful studies. I have to ask you a question about your collateral findings because they're the opposite of what we find. Our studies were in the Akita mouse model of insulin deficiency. But to first just ask you about the rat model of diabetes that you first showed, that's a genetic model?
It is multifactorial in these animals. They do have a defect in their insulin secretion from pancreas, but and they do develop insulin resistance at the beginning. We don't know exactly why they develop insulin resistance because they are not obese. But they are spontaneously diabetic. And I don't know if other, with the exception of one mutation in this insulin, in this protein involved in insulin secretion, I don't know any other defect. No.
The mouse model that I showed, I didn't show you the data, but for the loss of collaterals that we see with that diabetic model, it's a loss of beta cell activity. Do you see any changes in collateral abundance? Sorry, I'm focusing on collaterals, but I'm obsessed with them. Do you see any effects on collateral abundance before diabetes ensues?
No, we don't. Actually, when we looked at these animals, they start developing hyperglycemia around 6-7 weeks of age, and actually in 5 to 6 weeks they develop this pathology. So if you look at that age before the hyperglycemia goes up, we don't see these differences. So I think it's something hyperglycemia driven, and the severity of hyperglycemia I think is important, and it might explain the differences in the different diabetic models. So.
Thank you.
Okay. Hi, nice talk. So you mentioned in the diabetic mouse that the vessels were immature and that they have increased permeability. I'm wondering about the molecular changes to the junction molecules, or if there are other reasons that they're leaky. If there's more surface area because there's so many more vessels, maybe there's a constitutive permeability increases to the barrier change, or if the lack of pericyte coverage is somehow figuring into this.
I think it's both. If we are able to isolate, and of course we can debate that, endothelial cells from this particular animal, and then they are able to retain some of the angiogenic properties in culture, and they have harder time establishing a resistant basal resistance, for example. So they have some endothelial specific differences as well in terms of the barrier function. But we looked at occludin levels. It's, for example, that is lower in these animals. But other than, I mean, we did not manipulate those different either tight junction proteins or heterogeneous junction proteins, but we just saw a decrease in their protein abundance.
Okay.
But I don't know in terms of how it
Yeah.
affects the function.
Okay. It might be interesting if the parasites contribute to junctional changes.
Yeah.
I'm sorry, we're going to have to move on, so thank you very much.